It's a pleasure to moderate this next session. My name is Ralph Baric. I'm a professor in epidemiology in the Gillings School of Global Public Health. This session will focus on the epidemiology and the virology of the 1918 flu pandemic virus. We're actually quite fortunate to have the next four speakers, actually three that are in this session, one in the next, who are clearly rank among the top flu virologists in the world. They would be on anyone's top 10 list, I believe. The session will focus on the pathogenesis of the 1918 flu virus, talk about recent developments in vaccines, and the current status of anti-therapeutic compounds that are used to treat influenza virus infections, not only contemporary flu strains, but also avian and other potential pandemic viruses that might emerge in the future. We ask that you hold your questions to the end of the session. We'll have the speakers come back and sit at the table where they will take questions from the audience. It's hard to actually do justice to each of these next three speakers in this session. I've had the privilege to actually collaborate and work with all of them. They are great colleagues and wonderful friends. The first speaker is Dr. Adolfo Garcia Sastre. Adolfo, using sequence information in silico databases, resurrected the 1918 influenza virus at the beginning of the 21st century. Having access to that virus allowed us to begin to understand its pathogenesis, transmission, its susceptibility to vaccines and therapeutics. And so he's going to talk to us about this virus and how it causes disease. Adolfo is the Fishberg Professor of Microbiology and Medicine and Infectious Diseases. He's the Director of the Global Health and Emerging Pathogens Institute at the Icahn School of Medicine at Mount Sinai in New York. He's studied influenza viruses and other negative-stranded RNA viruses, as well as any other new emerging virus that appears in human populations for at least 20 years. He's made seminal contributions that are referenced throughout the literature. He's currently the director of the Center of Research on Influenza Virus Pathogenesis, which is one of the 5 NIH-funded centers on influenza virus research and surveillance. Our next speaker is Dr. Barney Graham. Barney is the Deputy Director of the Vaccine Research Institute at the National Institute of Health. In infectious diseases at NIH. He's the chief of the Viral Pathogenesis Laboratory, and he really oversees much of the research that focuses on the development of new vaccines against highly pathogenic respiratory viruses, as well as other emerging viruses. His research encompasses vaccine development for HIV, influenza, respiratory syncytial virus, coronaviruses, and other epidemic viruses that have emerged in the last decade. He is an immunologist and a virologist and a clinical physician, and I look forward to hearing the latest updates on the development of new flu vaccine strategies to control this important human pathogen. Our final speaker is Dr. Frederick Hayden, who is the Stuart Richardson Professor Emeritus of Clinical Virology and Professor Emeritus of Medicine at the University of Virginia School of Medicine. He has studied respiratory viral pathogenesis and clinical viral pathogenesis for many years, and he's been at the forefront of both developing and testing flu therapeutics that are used in human populations. And so he will give us updates not only on the current list of antivirals that are available for treatment of influenza virus infection, but he will also give us an update on things that are in the pipeline that will provide hope for future generations. So with that, Adolfo, please lead us off. Please welcome.
Thanks a lot. Thanks, Ralph. Thanks to organizers for having me here. It's really a nice mixture between public health and scientists, and I'm enjoying the first talks of this morning. So I hope also you like my talk that was already almost given by Ralph, telling exactly what I was going to say. So I was involved in the team that reconstructed the 1918 influenza virus, and I would like to tell the story about why we did that and what we have learned from this virus, and why we think it was important, the work that we did at this time, and how this virus still may actually teach us how fight against influenza viruses. So I have two slides that I like that saw right away the impact of 1918. Probably you are more familiar with some of them, at least with this one. This one shows the US life expectancy in the last 100 years. That has been increased due to modern medicine, antibiotics, hygienic techniques, et cetera, except for this big dip that happened in 1918. And this big dip was caused by the 1918 epidemic, was not caused by the Great War that was going also at this time. Great War had very little contribution to the dip that happened in life expectancy, not only in the United States, but in the whole world. The other is a picture from the Smithsonian. This is a picture of a little village in Alaska. And these are the picture of the survivors of the 1918 epidemic when the virus reached this remote village in Alaska. And what you can see from this picture is that there is no adults in the picture, and that's because all the adults died from 1918 infection. So although the 1918 was very dramatic infection in healthy adults, caused only around 2 to 4% deaths, but in remote areas, like in this village in Alaska, where probably there was no pre-existing immunity against influenza virus, it was even more devastating than in other areas. In this case, all the adults die of flu infection. Now, why to study this virus that was 100 years old now? Well, what the rationale that we have is that this virus contains determinants responsible for its success as a pandemic virus. And we don't have too many examples of pandemic influenza virus in humans. We have 3 examples, and there was also the 1918 that was there waiting to be recovered. Now, the virulence factors that this virus may have, why it was so much virulent to the pandemics, they were not understood at the time that we started reconstruction of the virus. And we thought that probably will unravel some new virulence markers that we don't understand it for virulence of influenza virus in humans. And finally, a new 1918-like virus in theory may evolve. So it may have been a single episode, but this one does not exclude the possibility that there is a virus that has similar characteristics that happens and come in the future as a pandemic virus. So we think that by knowing at least the determinants of virulence of 1918, we will have more information about what makes influenza virus more virulent in humans, what actually makes a virus a pandemic virus in humans, and how we can better fight against these viruses. So the problem, as you know, is that in 1918, it was not known that influenza was caused by a virus. There was no techniques to isolate the virus, and then the virus became extinct. The descendants of the 1918 virus continue propagating as seasonal influenza viruses in humans, but due to antigenic drift and evolution, the virulence of this virus changed and became less virulent. Now, while the virus was not available, what there was available was some sources of tissue where the virus was not there as an infectious virus, but there's still genetic material of the virus, quite degraded, but still pieces of the genetic material of the virus were there. And it was through our collaborator, Jeff Tauberger, at this time in AFIP, now at NIH, that he got access to material like that, and mainly he has been using 2 types of samples to sequence the genome of the 1918 virus. One is paraffin-embedded tissue samples from lungs of soldiers that died in 1918 of influenza virus infection, and they were collected as part of a biobanking of tissues from soldiers that die of unknown diseases that is going on for now many years. And the other was the body that was buried in one of these remote Alaskan villages, in this case Breivik Mission, of a person that died of 1918. As in the picture before, most of the adults died in this village. The army came and buried the bodies under the permafrost line, so the bodies have been frozen since then. there had been also deterioration of the tissue. Infectious viruses were not there, but with the help of Dr. Helting, he identified the grave and then got tissue samples to be sent to Jeff Taubenberger for performing sequencing. So we start with pathological specimens of people that died of 1918 of influenza virus infection, and then Jeff Taubenberger did sequencing with still not very sophisticated sequencing techniques that the ones that we have right now, but that was based mainly in PCR, PCR of small fragments, and little by little getting the sequence of the whole virus. And once that you have the sequence, then you can reconstruct the genes as plasmid DNA, and using what we call reverse genetics techniques, introduce the genes into influenza viruses, infectious influenza virus, and then ask questions about how the virus looks like. And here's an example of a single gene of the 8 genes of influenza virus that is included from 1918 into a typical seasonal human influenza virus. And the whole gene was reconstructed, and that was done at the end, the rescue of the virus by Terry Tampi at the CDC and the high biocontainment. And this is a picture, electron microscopy of the picture is indistinguishable from any other influenza virus. And just by looking to the sequence also, one couldn't say what could be the determinants of the virulence of this virus. There was nothing known at this time about the sequence of this virus that may have tell us why it was so virulent. So first, what's really a quite virulent virus? First, we use a mouse model of influenza infection in which we intranasally infect mice. And then if you do it with a human H1N1 from 1991, that they are descendants of the 1918 virus after many years of circulation in humans, you don't get, even if you use 10 to the 6 or more than 10 to the 6 infectious units to infect mice, you don't get lethality in mice. So this is mouse lethality 50. You need more than 10 to the 6 PFU in the mouse in order to get lethality with a seasonal H1N1 from 1991. Now, you introduce just 2 genes of the 1918, the hemagglutinin and neuraminidase, the surface proteins of this virus. Now you get a virus that is able to be lethal in mice with 10 to the 5, more or less, PFUs being required to get lethality in mice. You introduce more genes. These are M, NS, and MP genes. You don't change so much the lethality of the virus in mice. But if you make the whole 1918, then you increase lethality around 1 log and a half, meaning that not only the hemagglutinin and neuraminidase contribute to virulence, but also the polymerase genes, which are the ones that were introduced at the end, contribute to virulence. So the first thing we learned from these studies is that the virulence of this virus is multigenic. It's not a single thing that is responsible for the high virulence of the virus, which makes things at least a little bit give us more hope that it will be very difficult to get a virus like that, because you need to combine multiple virulence markers in order to be able to come out with a virus like that. So it's not a single determinant. Okay, this also can be seen here. If you infect mice with 10 to the 6 PFU of a H1N1 from 1991, seasonal human virus, then all of them survive. But if you do it with one that contains the hemagglutinin, neuraminidase, and 3 more genes from 1918, they all die. And if you get the 1918, the complete 1918, they die even more quickly than when they have, this is with the polymerases. Now, we just exchanged the hemagglutinin. So you have a virus that is like 1918, but with the hemagglutinin from the 1991, this virus, again, is not able to kill mice. So the hemagglutinin is important. As I mentioned before, the polymerase genes are important, as I mentioned also before. And if we look, so what happened to virus replication in these animals? So we look to viral titers. The one that is not lethal grows to around 1,000 infectious units in the lungs of these animals. This one that is lethal is around 10 to the 6. This one that is even more lethal, around 10 to the 8. And this one that is not lethal, around 10 to the 5. So it looks like, at least for 1918, replication is one of the major factors of virulence. If the virus is able to replicate to more than 10 to the 5 PFU in the lungs of these animals, then you get lethality. Now, in collaboration with Michael Case, we also looked to the host immune response that is happening in these animals. And we did that in a general way by looking just to the whole response and how many genes are coming up and down, which is seen here in this plot. So basically what one needs to see here, these are changes in gene expression from lungs in mice infected with the control virus and with different combinations of genes from 1918 or the complete 1918, day 1, day 3, and day 5. And the interesting thing to see is that in this part, which is the one that is infected with the total 1918, and that's a different mice, what you see is that there is more color. More color means that there are more changes, and more changes mean that there is a lot of inflammation in these animals. So a lot of host response, a lot of inflammation. Now, most of these changes are happening in genes that correlate with activation of immune cells, neutrophils, NK cells, macrophages, or T cells. And again, these changes correlate with the ability of this virus to replicate in the mice. The higher you replicate, the more changes you get. So what we think will happen is that high replication contributes to what it appears to be a deregulation of gene expression, a lot of changes, which is correlated with cytokine storm and immunopathology. And that's what we think is killing these animals. Now, independently, Yoshi Kawaoka, which is also here in the audience and will give also a presentation later on, his group also reconstructed the virus, and he found very similar things. Also, he performed studies in macaques, and then he found that even this virus is able to kill macaques, again, due to an aberrant host response, an exacerbated host response that occurs in macaques. Now, in our group, we used a ferret model, and the reason why we use ferrets is because they look a lot like humans, as you can see. So they behave like humans, at least with respect to influenza virus infection. They are naturally susceptible to influenza virus infection. That's not the case with the mice. And monkeys also, in general, they are seronegative to influenza virus infection. And they exhibit similar symptoms to influenza virus infection, fever, weight loss, loss of appetite, runny nose, sneezing, and respiratory symptoms. So with Teritampi, we put 1918 virus in ferrets, and we used 3 ferrets where the virus was intranasally inoculated. But in addition, we used another 3 ferrets, 3 contact ferrets, where they were not infected, but they were put in close proximity to the infected ferrets 1 day later after infection, allowing not direct contact between the ferrets, but allowing transmission by droplets and aerosols. So we are looking also to the ability of this virus to transmit in ferrets and what type of disease will be in the ferrets that are acquiring the disease, but not experimental intranasal infection, but by being infected by aerosols or droplets from infected ferrets. And the results were, again, high lethality. 2 out of 3 ferrets die in this control, in the one that are experimentally infected. And the contact ferrets, they were also infected, so the virus was transmitted from the infected ferrets to the contacts. And there was also quite severe disease, with 1 animal out of 3 dying in this group. And this correlated also with exacerbated infiltration of immune cells in the lungs of the ferrets that they were seeing in 1918, more infiltration of cells. That in control mice that we use also in these experiments for a virus that also transmitted well, but didn't cause severe disease, the seasonal H1N1 1991. Now, I told you about some of the genes that are responsible for that, but in order to see what actually are the specific genes responsible for maximal replication and virulence of the 1918, again in collaboration with Teritampi, we substituted every single gene of the 1918 virus with 1 gene of the seasonal H1N1. So we get a battery of viruses that are identical to 1918, except for 1 of the genes of the 8 genes that is now derived from the seasonal H1N1 from 1991. And then we look for this virus ability to replicate in human cells. And as you can see, most of them replicate quite well, except for these 3 here. And these 3 are the ones that contains the hemagglutinin, the neuraminidase, and the PD-1 genes. So again, multifactorial, the ability of replicate to high levels in human cells, and you require at least HA, NA, and PD-1 in order to get these high levels of replication. And if you put this in mice, all of them are lethal, except the ones that have less lethality, so don't kill all the mice, the one with the HA gene, the one with NA gene, and the one with the PD-1 gene. Again, correlating high levels of replication with ability with lethality and virulence in mice. Now, Jeff Taubenberger not only sequenced the hemagglutinin, that was one of the genes involved in virulence, from the Breivik mission isolate, but he also sequenced hemagglutinin from 2 soldiers that died in 1918, one in South Carolina and one in New York, and also from 2 London samples that he was able also to obtain from tombs in London that they have leather, where the bodies were buried in leather cases. And the hemagglutinin was identical in all the cases, except for one change here in amino acid 225 from D to G. So there were 2 different polymorphisms in the hemagglutinin of 1918 from these sequences. And the interesting thing is that this amino acid has been known to play a role in receptor specificity of the hemagglutinin. So the hemagglutinin binds to the receptor before entering. The receptor is sialic acid, but sialic acid can come in 2,3 and 2,6 linkages. The avian hemagglutinins from avian viruses, they have usually these 2 amino acids, H1s, that bind to 2,3 linkages that are present in avian hosts mainly. And the human ones, they are like this, and they bind to 2,6, which are the ones that are present in the human host. Now, we will predict that by this change, you may change receptor specificity of the hemagglutinin, and even if you introduce a new change here, then you will change completely receptor specificity. So we then use the wild type, the one that we used before, the Breivik mission of South Carolina. Then we also constructed the virus with a single change, and a virus with what we call avianized that contains these 2 changes in the hemagglutinin sequence. And then we look for ability to bind to receptors, 2,3 or 2,6. So in red is binding to 2,3s, in blue is binding to 2,6. The avian hemagglutinins bind mainly to 2,3, the avian receptor. The human hemagglutinins from Moscow or from the South Carolina isolate of 1918, they bind mainly the human receptor, the blue receptor. Now, the New York, the one that has the single amino acid change, has dual specificity for receptors, and 1 more amino acid change now make it specific for 2,3, not anymore for 2,6. And this is interesting because in addition to the receptor being present more in birds, 2,3 and 2,6 more in humans, 2,3 is also more abundant in the lower respiratory tract of people, where infection is usually more severe if the virus goes there. So then we use these viruses to infect, again, ferrets, and there we use, when we use the previous, this was the previous virus that we used, the control and the 1918. Now with these avianites, they die, the ferrets, but this one does not transmit. And that was one of the first evidence that receptor specificity is important for transmission of the virus, and it was later on confirmed by Joshi and Ron Foussier using H5N1 viruses, that they need to have the right receptor specificity to be able to transmit among some other things. Now, the other thing why we reconstructed this virus, we wanted to make sure that we can fight against a virus like that. So for that, we have 2 types of antivirals right now, M2 inhibitors and NA inhibitors. So are influenza antivirals effective against the 1918 flu virus? If we use a lethal challenge of mice that are given oseltamivir, neuraminidase inhibitor, we prevent severe disease, so the animals survive. And if we use amantarin, we also can prevent lethal disease, and these are using some control viruses that are resistant to amantarin that are not inhibited by amantarin. Now, I showed you that it has a lot of inflammation, so can we control inflammation just by reducing inflammatory cells? So these are the amount of inflammation that you get in the lungs of these animals as compared to a control virus. If you deplete now neutrophils and alveolar macrophages, which are the main inflammatory cells going there, you actually don't reduce mortality, you actually increase mortality. So although inflammation probably is a good way how to get into this virus for treatment of viruses with severe disease, you cannot just completely reduce inflammation, because if you do that, then you get a worse outcome. Now, how about vaccines? Vaccines, if we use a vaccine that is based on the hemagglutinin neuraminidase of the 1918, this is protective. So this is good also. The vaccines will be able to work. This, with NH3 and 2, this obviously doesn't work because it's a completely different subtype. But even vaccines that are very far away with the 1999, they are partially protective. And as you get up, then you get closer antigenically to the 1918, and then you get better protection. So the closer you are antigenically, that's something that we knew already, the better you are. But at least vaccines are also working against viruses like 1918. So what do we know now about this virus? So it's the only known human influenza virus lethal to mice, ferrets, and macaques. Glycoprotein and polymerase genes of the virus contribute to enhance virulence by promoting viral replication. Alveolar macrophages and neutrophils have a protective role and probably also an exacerbated role in the disease. Single amino acid change in HA changes receptor specificity and determines transmission in ferrets. But the good news is that viruses containing 1918 genes are sensitive to existing antivirals, and H1N1-based vaccines are protective. So if we ask the question, would the 1918-like H1 viruses be today as lethal as in 1918? If it will be the exact virus, the 1918 virus, the good news is that the new pandemic H1N1 virus is antigenically very related, and vaccines that we have now used against the new seasonal H1N1s based on Calo9, they are not only inhibiting the seasonal H1N1s and the old pandemic 2009, but they are also inhibiting the 1918 virus. So most of us have now antibodies against the 1918 virus. So in order for a virus like 1918 to be as virulent, it needs to have a different hemagglutinin than the one that it had. And just to say, there was a big group, the one that used these studies. These are some members of the group. On interest of time, I'm not going to name the people. I named some of the people before. And that was done with the support from NIAID and the Keck Foundation. Thanks.
Good morning. It's my pleasure to be here. I thank Ralph and for inviting me and to the organizers for allowing us to share some of our work. I'm going to go through what some of the options are for universal flu immunization and trying to find vaccines that are better than the ones we already have. So I work at the Vaccine Research Center on the campus in Bethesda at NIH. We're in this building 40. Give me just a minute. Here, and these are the principal investigators and program heads that I work with every day. The VRC was founded to work on HIV infection and develop an AIDS vaccine, but in the meantime, we've taken on a number of these other projects, many viruses that you've heard of coming over the last 4 or 5 years. Much of the technology that we're using to address these other virus infections has come as a result of funding for HIV and AIDS. So just as the 1918 flu epidemic, I think, stimulated some funding and investment, for instance, by the Rockefeller Foundation, I think the AIDS epidemic has stimulated investment in biology and immunology that has resulted in some of these projects that I'll briefly tell you about. We use a number of different delivery platforms demonstrated here, either gene-based delivery of a vaccine antigen, making virus-like particles, or making protein-based vaccines, either as soluble proteins or mounted on nanoparticles. I'm going to tell you some stories today about a flu program based on this nanoparticle. And we also make monoclonal antibodies that can be given passively. And in addition to the research program, we have a process development group of chemical engineers that help develop ways to manufacture vaccines. We have a manufacturing pilot plant that can make GMP-grade product for use in human trials, and we have our own phase 1 clinic. We do a good laboratory practice for sample analysis, but as we advance into phase 2 and higher phase trials, we need partners. So the reason we need a universal flu vaccine, I think you've heard this before, but we're still using 70-year-old technology. We still grow viruses in eggs. We still inactivate them in about the same way. And on good years, it's about 60% effective, and in bad years, it can be as low as 10% effective. I think it's fair to say it's still better than not getting the flu vaccine, but I think people argue that it's marginally better in some years. We have to remake it every year. We have to have 2 meetings a year to prepare for southern and northern hemisphere outbreaks, and it's generally not going to be effective against new pandemic strains. So what are we going to do about that? The reasons it's so difficult are primarily the antigenic and genetic variability of these viruses from year to year, either through drift, as you just heard Adolfo describe, or through shift, which happens when you get whole new gene segments into the virus. So this level of antigenic variation is only seen in a few other virus diseases like HIV and hepatitis C, for instance, but this is a major problem for flu. Another problem we face in immunizing for influenza is pre-existing immunity. And I'm going to get into some immunology, B-cell immunology, a little bit at the end, showing you how pre-existing immunity is a problem for inducing new responses, but it also gives us an opportunity to identify responses that could be more effective in the future. And then the other problem we have is that flu is usually most severe at the extremes of age. And so immunizing young infants and immunizing the elderly have their own problems. So there's many targets. This is a cartoon of a flu virus with its gene segments inside, a lipid envelope, and the surface proteins include the neuraminidase or the hemagglutinin. And there's many gene targets or antigenic targets on the virus. I'm going to focus most of my attention on the hemagglutinin molecule today and give you some examples of new technologies and how they may be useful. So as you've heard, we're facing not only flu pandemic threats, but pandemic threats from many other things because of the way we've disrupted our ecology. And the question is, can we turn vaccine development into more of an engineering exercise by taking advantages of some of our new technologies to develop different approaches, better approaches than the traditional approach of empirical trial and error type of approach to vaccine development. And I feel like I'm a little bit of risk here of becoming a caricature of the outbreak narrative because I'm going to get into some technical details. So I'm going to be the technologist who thinks we may have solutions here. Just to make it a little more personal, let me tell you a story of how I'm connected to the 1918 epidemic. My grandfather was an osteopath in Pratt, Kansas, which is not too far from Haskell County, where John Barry at least thinks that maybe this 1918 flu emerged. And his first wife died in 1918. So if he had not married my grandmother a few years later, I wouldn't be here talking about 1918 influenza. So I have mixed feelings about the 1918 flu outbreak. So I'm going to give you an example of how structural biology has informed our ability to now make new types of vaccines that have more specificity. I'm going to tell you a little bit about how high-throughput sequencing can identify not just the viruses that emerge, but also the antibody sequences and responses that we make to viral infections that can inform our targets for vaccine responses. And then how rapid isolation of human monoclonal antibodies that's really just happened over the last 8 to 9 years has really changed everything in how we think about vaccine design. So RSV is a childhood virus primarily. It infects young infants. The peak age of hospitalization is about 2 to 3 months of age. It infects the small airways. It causes lots of hospitalizations globally. It's not as lethal as influenza, but In the elderly populations, it actually has nearly as much excess mortality as seasonal influenza on a year-to-year basis. And so it is a substantial respiratory infection and it's the leading cause of hospitalization in children under 5 globally. So on the surface of RSV, it has a fusion glycoprotein. It's very similar to the hemagglutinin of influenza. It's what we call a class 1 fusion protein. And you can see if you do cryo-electron microscopy on the surface that there's both a short and a long form of this F protein. And for the last 40, 50 years, we've been using this post-fusion form of the protein as a vaccine. And about 5 years ago, we were able to solve the structure of this pre-fusion form before it rearranges. And it revealed some neutralization-sensitive sites on the protein that had not been known before when we were just using this protein. And so by identifying the structure of the functional form of this protein before the rearrangement occurs and identifying neutralization-sensitive sites, making a vaccine, we could show that it's much more immunogenic, and it's now in a vial in early clinical trials, and indeed it's inducing responses that are 16 to 20-fold higher than baseline instead of 2 to 3-fold higher than baseline from these other previous failed efforts. And we think it's one of the first clinical proof of concept for structure-based vaccine design. Knowing the atomic level structure of a protein and understanding the exact target of neutralizing antibodies can really change everything. So can we apply this new technology to flu? So I'm showing you another cartoon of the flu virus and the hemagglutinin molecule. This is my friend Ian Wilson, who this is one of the first glycoproteins ever crystallized and solved. And of course, Ian didn't have many computers back then, so he had to make a stick and ball model of his hemagglutinin molecule. And he's standing here by it. And so we've known for a long time the atomic level structure of the hemagglutinin of influenza. I'm just going to show you a brief movie to give you context or orient you to what I'm talking about. The hemagglutinin is this trimeric protein, and it has a head region and a stem region. It's blocked or protected by sugar molecules or glycans that you see on the surface, and most of the changes from year to year happen on these red spots on the head. But there are sites of vulnerability, either one in the stem here or one in the sialic acid binding pocket here at the top. And there are known monoclonal antibodies that can bind either the head region across strains or the stem region across strains. And the question is, can we make vaccines that can elicit these antibodies that can cross-react across subtypes and maybe even across groups of hemagglutinin molecules? So there's lots of ways to use this hemagglutinin in a vaccine. You can express it in live viruses. The way we make the conventional vaccine, as I said, you grow a live virus in eggs, you disrupt it, and you harvest whatever proteins are left over and then give it as an inactivated product, which is mostly hemagglutinin. You can also make virus-like particles that don't have the flu genome. You can deliver it with nucleic acid or gene-based vectors. You can give it as a soluble protein, either as a normal molecule or, as Adolfo suggested, maybe a chimeric head molecule. I'll describe that in a minute. Or you can use protein engineering and design vaccines that are either based on just the receptor binding domain, the full-length hemagglutinin, or just the stem part of the hemagglutinin, display it on a nanoparticle, and ask, can you do better than natural immunity? So I'm going to focus primarily on this approach and give you some examples of what we can do. The reason is that if you look at the stem structure and you look at the sequence of the hemagglutinin, you can arrange or divide the hemagglutinin molecules, there's 18 of them that birds all carry, into two major groups. And seasonally, we are exposed to the H1 hemagglutinin, or the H1N1 virus, the H3N2 virus, and the 1918 was this H1. We're worried about avian flu, H5 or now H7, that sometimes shows up in China that are largely transmitted from avian sources. And if we look at the stem region in more detail, you can see that the antibodies that can neutralize based on stem binding can occupy two different types of footprints. One that can get the group 2 viruses, one that can get the group 1 viruses, and some that can cross both by slipping in between these two sugar molecules. So if you don't understand that geography on the protein, you won't be able to make a vaccine that can elicit those responses. Now again, just to orient you, this is a plasma cell. This is what makes our antibodies. It lives in the bone marrow. These are antibody molecules, and this is a flu virus. So one question is, if you make antibodies to the stem, can they actually access the virus and get to the right place on the hemagglutinin and do something to block the virus? This study by Audrey Harris a few years ago showed that indeed an antibody that binds the hemagglutinin here, and you can see them demonstrated here in a cryo-electron microscopy study, they do access the stem region. So if we could induce these stem antibodies, could we make a better vaccine for influenza? Those antibodies are not as potent as the ones to the head, but they're much more conserved between strains. So here's an example of how we've addressed this. This is the full hemagglutinin, and if you remove the head and do some what we call circular permutations to tie the top of that head together, you can stabilize the stem trimer by adding additional molecules and mutations inside to pack it and hold it together from the inside. Then you can mount those trimers on what's actually a self-assembling molecule called ferritin. So you can have now 8 trimers of the stem on a ferritin molecule. That's the group 1 approach. For group 2, it was much more difficult, and it's taken actually about 5 or 6 years to gradually achieve a stabilized molecule that has the right exact atomic level structure. So by making these a variety of mutations, you can eventually get to a place where for the H3N2 type viruses, you can have a similar type of vaccine. Now, I'm going to go back now to what I said about sequencing and antibody lineages. We've done some studies with both H5 vaccinations and H7 vaccinations, which humans don't have so much pre-existing immunity to. And by using some of these proteins now as a probe in a flow cytometry device, which can monitor and measure proteins on individual B cells going through a laser detection system, you can ask whether you can find B cells that cross-react between H5 and H1 within group 1 viruses. They would be in this upper right quadrant. Each one of those dots represents an individual B cell. So you can use the hemagglutinin probes to find these kinds of B cells. You can also find B cells that cross-react between group 1 or H5 and an H3 from these people who were vaccinated with pandemic strains. And what you find is that there are certain types of antibody lineages, meaning the heavy chain sequence that is used to make the antibody. For instance, to group 1 cross-reactive antibodies is almost all VH169. It's very heavily biased toward this type of an antibody. The ones that see cross-group responses are much more diverse and complex, but you can identify antibodies. You can even identify motifs in the sequences of the antibodies that recognize the stem in a cross-reactive way. So we now have about a dozen of these types of lineages that we know can recognize both groups. And we find that immunizing with the group 1 viruses tends to stimulate these VH169 type responses that are more group 1 oriented. But immunizing with H7 or a group 2 virus, more similar to the H3, we induce a number of antibody lineages that have the capacity for cross-group neutralization and also cross subtype neutralization. So we have made these types of products. We've shown that they can protect ferrets. If you use an H1 type of vaccine, you can protect against a lethal H5 challenge, as shown here. We've also now made the group 2 on both a ferritin and a lumazine synthase type nanoparticle. And since we know these sequences of the antibodies we're trying to target, we can go to what the germline version of those. That's what's actually encoded in your genome before it mutates to become more effective. And so we can put the germline versions of those antibodies into Ramos cells in vitro in a tissue culture flask and ask, do our antigens recognize, or do those germline versions of antibodies recognize our vaccine antigens? And indeed, we can make now group 2 vaccines that can stimulate the germline versions of antibody lineages that we know have the capacity to become broadly neutralizing antibodies. So these kinds of technologies have created a whole new philosophy and way of designing and also tracking and then measuring responses to vaccines. So I just said that. We've also, I just want to make sure it was clear that we've defined these structurally defined sites of vulnerability and specific antibody lineages that can now be targets for flu vaccines. We don't know if we can induce them in people who are pre-immune. It may have to start back at the level of the naive infant, but we're going to know that within the next few years. And we have designs based on full-length HA, the stem, or the RBD. I haven't been able to tell you about all those, but you see that there's a lot of new options and ways of combining these technologies to get to a better flu vaccine. And just to finish, I want to just tell you how we're taking all these technologies back to work on HIV, because HIV is probably the most difficult vaccine target. It has all these ways of evading neutralizing antibodies, but by combining our work on RSV and flu and coronaviruses, we're learning more now how to make better HIV vaccines. So I'm going to thank the people that helped me with this work from my lab and from the VRC. John Moscola is our director. Adrian McDermott is an important collaborator. Sarah Andrews works in Adrian's lab. Gordon works in John's lab, and I don't have Jeff Boynton on here, but he helped me make these movies, and Jason McClellan was involved in the original RSV structural discovery. So thank you very much.